Imagine you're standing on the deck of a research vessel off the coast of Iceland. It's 2006. The water is gray, the air is cold, and a team of marine biologists has just pulled something up from the ocean floor, a clam. Unremarkable, palm-sized, covered in sediment. They crack it open to count the growth rings on its shell, the same way you'd count rings on a tree. And then, someone does the math twice, because the first answer can't be right. 507 rings. This clam was born in 1499. It was alive when Leonardo da Vinci was still painting. It survived the Black Death's final European waves, the entire reign of the British Empire, 2 world wars, and the invention of the internet. It was filtering seawater when Shakespeare was writing plays. And it died not from old age, but because the scientists studying it didn't realize what they had until after they opened it. Here's the question that doesn't go away. If evolution optimizes for survival, why did it give a clam 507 years and us only 120? The answer isn't one thing. It's never just one thing. There's a biological countdown ticking inside every cell you own. There's an animal so small you could lose it in a raindrop and it may be genuinely immortal. There's a cost to living long that evolution made every species pay. And there's a reason humans specifically got the short end of this deal. A reason that turns out to be calculated, deliberate, and stranger than you'd expect. Let's go. People assume animals age the way cars wear out accumulated mileage, friction, entropy. Use a thing long enough and it breaks down. That's not what's happening. In 1984, Elizabeth Blackburn and Carol Greider at UC San Francisco discovered telomeres, protective caps on the ends of every chromosome, like the plastic tips on shoelaces. Every time a cell divides, those caps shorten by 25 to 200 base pairs. When they get too short, around 3,000 base pairs, the cell stops dividing entirely. It goes into senescence, and it starts releasing inflammatory signals that begin breaking down surrounding tissue. The whole body follows. Blackburn won the Nobel Prize for this in 2009. Your cells aren't running out of fuel. They're receiving a shutdown command encoded at the molecular level. Now here's where this gets strange. Naked mole rats, small, hairless, deeply strange rodents maintain their telomere length far longer than mice of identical body size. Mice live 3 years. Naked mole rats live 37. Bowhead whales carry specific mutations in their DNA repair genes that keep their chromosomal caps stable for over 200 years. Human centenarians, studied in the Okinawa Centenarian Study, show measurably longer telomeres than age-matched peers who died decades earlier. The length of the fuse determines the length of the burn, and the enzyme that rebuilds the fuse telomerase is switched off in most human adult cells after development, deliberately, because active telomerase in adult tissue dramatically increases cancer risk. The clock inside your cells isn't a flaw, it's a trade. Your cells aren't dying because they can't repair themselves. They're dying because they were told to stop. Now let's talk about Ming. The ocean quahog clam, Arctica islandica, lives across the North Atlantic floor, and populations off Iceland routinely average 150 to 200 years per individual. Ming hit 507 because its mitochondria run what biologists call cold and clean. Extraordinarily low levels of reactive oxygen species, the metabolic byproducts that damage proteins and DNA. Paul Butler at Bangor University confirmed Ming's age in 2013 using carbon-14 isotope dating cross-referenced against shell ring counts. Ming's tissue showed no meaningful signs of cellular aging compared to 100-year-old specimens of the same species. Red sea urchins off the Pacific coast show the same pattern 200 years. No measurable increase in mortality rate with age, a phenomenon called negligible senescence. These animals aren't fighting aging. They're barely generating the damage that causes it. A creature with no brain, no spine, and no ambition quietly outlived 15 generations of human civilization, and we didn't even notice. Here's something else that almost never comes up. A Greenland shark named Nothing, because no one names them 5.02 meters long, caught in the North Atlantic, was dated in 2016 by Julius Nielsen at the University of Copenhagen. He used radiocarbon dating of eye-lens proteins, which form before birth and never regenerate. The result? 392 years old, give or take 120. That shark could have been alive in 1505. It was certainly swimming during the 30 Years' War. Greenland sharks grow at roughly 1 centimeter per year, the slowest growth rate of any vertebrate. They live in water between -1 and 10 degrees Celsius. Their metabolic rate is the lowest recorded mass-specific metabolic rate of any shark species. Cold slows oxidative stress. Cold slows cell division. Cold extends every window before telomere erosion reaches its threshold. The oldest vertebrate on Earth never saw sunlight and has been hunting in the dark since before your country existed. Here's the part that almost nobody talks about. Every mechanism we've just described, telomere maintenance, low oxidative stress, metabolic suppression, we understand them. We've mapped them at the molecular level. We've replicated some of them in laboratory conditions, so why haven't we applied any of this to ourselves? Either fixing human aging is genuinely impossible, or the biology is not actually the hardest part of the problem. Hold that. First, the strangest case. Turritopsis dohrnii, a jellyfish, 4.5 millimeters long, native to the Mediterranean, can revert its cells to their embryonic state after reaching adulthood, fully, systemically. Every cell in its body converts back to a totipotent stem cell. It restarts its entire life cycle. Shin Kubota at Kyoto University has kept colonies alive through this reversal, more than 10 times in laboratory conditions. Wild populations have spread through every major ocean via ship ballast water. Its close relative, Turritopsis rubra in New Zealand, shows partial reversal, suggesting the capability evolved on a spectrum. No mammal can do this. Differentiated mammalian cells have lost the epigenetic flexibility required. In exchange for a nervous system, a circulatory system, a skeleton, a brain evolution, stripped out the reset button. It doesn't cheat death. It just refuses to take aging seriously. Now, let's tackle the biggest myth here. Longer life is not free. João Pedro de Magalhães at the University of Liverpool analyzed maximum lifespan data from over 4,000 species in 2013. He found a near-perfect negative correlation. Every doubling of maximum lifespan corresponds to a 30 to 50% increase in age at first reproduction. Greenland sharks reach sexual maturity at 150 years and produce few offspring per cycle. Bowhead whales live 200 years and give birth to a single calf every 3 to 5 years compared to dolphins, who live 70 years and give birth annually. Naked mole rats live 10 times longer than similar-sized rodents, but have suppressed immune inflammatory responses so dramatically they can barely fight off fast infections. Thomas Kirkwood at the University of Newcastle called this the disposable soma theory in 1977. Organisms have a fixed energy budget. Energy spent on cellular repair is energy not spent on reproduction. Evolution in high predation environments always chooses reproduction. Longevity only wins in the deep ocean and the Arctic, where predators are rare and patience is the only strategy available. A long life and a fast life are the same energy budget, just spent in different directions. Which brings us to humans specifically. Jan Vij and Zhao Dong at Albert Einstein College of Medicine analyzed supercentenarian mortality data from 40 countries spanning 1968 to 2006. Maximum human lifespan plateaued at roughly 115 years in the 1990s and has not moved since, despite antibiotics, surgery, vaccines, and modern nutrition. The ceiling is genetically encoded. Jean Calment of France, born 1875, died 1997 at 122 years and 164 days. The oldest verified human life ever recorded remains the outer boundary, despite a global population 8 times larger than it was when she was born. Peter Medawar explained the mechanism in 1952. Natural selection cannot eliminate mutations that only cause harm after reproductive age. A gene that triggers cancer at 80 was never seen by evolutionary pressure. Almost no ancestor survived long enough to express it. Evolution didn't give you 120 years because it cared about you. It gave you 120 because it never had to care past 40. There's one last thing I saved for the end. Every civilization-level achievement humans have produced: language, writing, science, democratic institutions. Accumulated scientific knowledge runs on a specific generational rhythm. Ideas form in one generation. They get tested by the next, revised by the third, institutionalized by the fourth. Ancient Greece, the Islamic Golden Age, the European Scientific Revolution, the modern Silicon Valley, all of them operate on roughly the same 0 to 8 0-year generational cycle. That rhythm requires death. A civilization of 500-year-old humans would be one where the people who made decisions in 1550 were still making them in 2025. The same mechanism evolution used to limit your lifespan is inadvertently, one of the structural preconditions for a civilization capable of progress at all. Altos Labs raised $3 billion in 2022 to reverse aging. Google's Calico project has been running for over a decade. David Sinclair at Harvard is working on NAD+ and sirtuin pathways that may extend healthy lifespans substantially. These are not just biological experiments. They are, without saying so openly, experiments in civilizational structure. If they succeed, inheritance law, democratic elections, institutional leadership, and long-term planning will all need to be redesigned from first principles. The biology may turn out to be the easier problem. And so you're back on that research vessel. Cold air, gray water, a palm-sized clam with sediment on its shell. Ming was born in 1499. It survived the Renaissance, the Scientific Revolution, the Industrial Revolution, 2 world wars, and the moon landing. It filtered seawater. It added 1 ring to its shell each year. It didn't know about Shakespeare or Newton or Hiroshima. It just continued quietly, efficiently, for 507 years, until the morning a team of scientists pulled it up from the ocean floor to try to understand why it lived so long. They opened it to find out. The oldest living thing we ever found, we killed it trying to figure out why it wouldn't die.